Hi, I'm Meredith Blackwell, and I'm here with Tim James. We're at the Mycological Society of America meeting at Minneapolis, Minnesota. The year is 2019. And I want to talk to Tim about his work, starting out actually, though, with when he was born, where, what sort of family he was born into, were they supportive or not, all that kind of thing.
Okay.
So we can get an idea of the background of many mycologists that we talk to. So,
Right.
Tim.
Okay. I was born in Georgia.
Where,
I-
small town or?
I grew up mostly in a town called Stone Mountain.
I know
and
it.
You should know it if you're in the South.
Yep.
It's it's got a huge granite outcrop, and on the side of the outcrop.
It's called a monadnock.
Monadnock. Okay, that's the term for it.
I think so. These granite outcrops that stick up when everything else gets planed down. At least I think I learned that in geology.
Yeah, it's in a massive hump, and it has a lot of history there. But I won't go into that part of it.
Yeah.
And so I had just a normal childhood. My father's from Tennessee, my mother's from Korea, and just had a regular kid life.
Was that unusual, Koreans in the population?
It isn't anymore,
Yeah,
but back
but at the
then,
time.
certainly there were very few Koreans, other Koreans.
Yeah.
I was always interested in science, math, and there's no particular reason that I really ended up as a mycologist, as far as I can tell. I was, fascinated with ants and running out in the woods, fishing, that kind of stuff, gardening, when I left for college and went to George Washington University for 1 year and was studying journalism.
In D.C.?
In D.C.,
Yeah.
right. So I'd gotten into sports journalism of all things, and
Did you play sports?
not well.
But you wanted to.
Yeah, I love sports. I love everything about it, but I'm kind of not tall enough to do like the basketball that I love to do. But anyways, so but after a year, I kind of lost interest in that, and ended up kind of going back to Georgia, and went to the University of Georgia, and kind of not really knowing what I wanted to study, but realizing that journalism really, especially sports journalism, wasn't kind of the contribution to society that I wanted to make. And on a lark, kind of went into botany, thinking that, well, it's pretty clear that human history and the future is inextricable from plants, and that they're going to be, proper tending of plants will be essential for the future. And then by chance, I'm just kind of going into the narrative of
No,
this,
that's what I want to know.
So I'm just maybe 20 years old or something, and I'm studying botany, and there were very few of us, but one of the remarkable things is that there was a guy there named David Porter, and he was a mycologist, and he is the one who kind of got me really interested in mycology.
I didn't know that.
Yeah, it was that, and taking Charles Mims' class, and we had Tony Glenn as the TA. So those three remarkable scientists are the ones who encouraged my interest in mycology.
Interesting. Yeah, and Charles is a great friend of mine, and we've collaborated a lot. And so I'm glad to hear you mention. He'll be happy to hear that. He walked out, and that was the end of mycology. He won't even talk about it now.
Yeah.
But he put his whole heart into it.
Oh yeah, he was a remarkable teacher.
Yeah.
Everyone who took his class will remember him forever.
Were you there when Mel Fuller was there?
I
as
was
the chair?
there, yeah. I don't remember if he was chair or not, but he was just close to retiring. And he did come into the fungus part of the botany class and taught that. And I also remember thinking that was really exciting. And so Georgia has a lot of mycology there, and they have an emphasis on zoosporic fungi,
Right.
actually. So
that was your start in mycology, wasn't it?
right. And David, you know, he was interested in mushrooms, and he was interested in everything, and he's, you know, remarkable knowledge of mycology.
Yeah, I actually remember him with some of the marine grass parasites, the labyrinthids.
Right, the seagrass wasting
Yeah.
disease.
Yeah, that's what I most remember him for.
Or eelgrass wasting
But
disease.
also, one of my friends Daniel Hink's mother and his aunt actually
Right.
worked with him.
Right.
Did you know that?
I did know
Yeah,
that.
she worked
I
on
never
or not hallucinogenic, but bioluminescent mushrooms.
Right, yeah.
So he did have a very wide breadth of information about fungi. Yeah. And I think when Mel was there as chair, he got people in to teach freshman biology and other areas. Wherever he could fill it with the mycologists. So I don't know if any of the others were around, but you were probably past the stage of having a mycologist for freshman biology. But anyway, yeah. So after Georgia, did you do undergraduate research there?
Right, yeah, I did. And the first thing I did was work in a plant population genetics lab, a professor named James Hamrick.
Yeah, I've known him.
Yeah, he's into tropical tree population genetics, and I was working in his isozyme lab, basically mostly grinding plants up, extracting proteins for analysis, and making buffers and whatnot. And I think probably by my senior year, I wanted to do a research project. And I kind of wanted to do it on fungi, and also to use the population genetic tools that we were working on.
But you'd already been interested in fungi then, even
Right.
over the plants.
Yeah, so when I was tooling around in David's lab and just, you know, learning about fungi, and I thought, you know, I mean, when you take a class like Charles Mint's class, you know, you fall in love immediately, and the microscopy part of that was really exciting. But at any rate, I wanted to have like a population genetics project. And so I think what ended up happening is David Porter had done a sabbatical with Ritas Vilgelis and at Duke University. And Ritas kind of hooked me up with this very interesting project on Schizophilum communi. And he had this global sampling regime that he had done on his sabbatical. And the question was basically, what is the global population structure of this really, really common mushroom? And you know, Ritas at the time had been going and looking at things like oyster mushrooms and discovering that there was this deep geographic structure. And the question was kind of like, in this species, which is really common, reproductively, a single species, is there any population structure? And
So you were still at Georgia, or had you gone to work at Duke?
I was still at Georgia.
Oh,
yeah.
so he was helping you.
Even back then, yeah. So, and it was something I did maybe over the summer, one summer. Yeah, and it was pretty cool. Immediately, it was obvious there was structure, actually, even in Schizophilum communi.
Yeah, I've always wondered about that. I guess I should read more of your work. But I've read
back.
your other work, but not that far back. But Baccharis, which is a conch genus that's very, very common. I have never seen it along the Gulf Coast when it didn't have Schizophilum on it.
Wow.
Yeah, you just go for miles in the wetlands in South Louisiana, and it's there all over the place. And I've always wondered about it because it seems so specific on that.
On living.
Living ones along the roadside. But I've been with Ritas when he held a plate out as we're driving across the Atchafalaya basin in Louisiana, and that was kind of interesting, trying to pick up stuff. And I never thought that his baited plate would pick up opposite mating types of something that was
Right,
there, but
yeah.
it apparently works, huh?
Yeah,
Did
it
you do
does-
that?
I did do that also with
Mhm.
Schizophyllum
And it worked?
community. Yeah, remarkably well.
Interesting.
So, the spores are everywhere.
Yeah. Interesting. So, you finished up your project. Was any of it published
Right. We did publish one paper in evolution
as an
from
undergraduate?
that.
That's
Yeah.
pretty neat.
Yeah. I mean, it took a while.
Mhm.
I was a grad student already. Things took a
Mhm.
lot longer back then to publish.
When we also, I think we didn't, at least I didn't use email much until much later after I'd started. But I realized later how much it helped to have email and not have to do
Right.
snail mail, especially if you were publishing in Europe or something.
Yeah.
So, anyway.
And
So-
there's many improvements for not having to print out the 3 copies
Yeah.
and send them
Yeah.
by snail mail.
Proposals too. You had more copies to print out. So anyway, so then you decided to go to Duke because of Rita's. He had helped you.
Right. Yeah, exactly. I was interested in mushroom genetics, and kind of, the ideas I had when I started were pretty vague. And I ended up working on the evolution of mating type genes in mushrooms. We were kind of interested in understanding like what genetic changes underlie mating system evolution when you go from, say, tetrapolar to bipolar, and bipolar to homothalic. We never figured that one out, and actually that's still an open question in mycology in mushrooms.
And so your early experience with all the zoospore people in Georgia, did that help you later?
Right, yeah. Well, I mean, I got going on zoospore fungi even going back to Georgia one time. So, yeah, I was trying to think about when the first time I met Joyce was, but Joyce Longcourt,
Yeah.
that is,
Yep.
who I've collaborated with since probably 1997 or something. But there was a sample. So David Porter was really into Kittredge, and they're marine fungi mostly.
Right.
So I had a sample that I asked a friend to collect from Mono Lake in California, and I was going to isolate.
That's a big salt lake.
It's a big inland salt lake,
Yeah.
And so she brought this sample back, and then I was trying to isolate Kittredge from it, and I ended up isolating a Kittredge from it, and that was interesting. So it's like, yeah, this is a Kittredge that likes hypersaline conditions, and then I think that's how David said, well, let's talk to Joyce, and so we as a group, I think as a lab group, also had Celeste Leander in that group, and Sharon Mosley was coming into his lab. We started this Kittredge systematics project.
And so had the Pete grant been funded at that time, or was that a little later?
I'm a little fuzzy on the timing.
Okay.
I think
I don't remember what
maybe
year it
starting
was.
when I was leaving.
Yeah. But that really helped develop Kittredge biology.
Yeah.
So when did you get interested in the frog Kittredge and Batrachochytrium?
Well, because I was doing systematics, and I mean, honestly, it's kind of just because there were so few people working on Kittredge, and I had this good collaboration with Joyce that she asked if I would be interested in working on the cultures she had to understand whether the fungus was undergoing sexual reproduction. And so we were celebrating John Taylor at this meeting, and John had made many contributions to understanding whether a fungi were sexual or not, and we were basically going to use that kind of approach. And this is maybe 2001, 2002. And it turned out we couldn't really use that approach because the fungus was extremely low diversity, and it also turned out to be diploid, which we were not expecting.
Were these U.S. samples, or were they from other places
There
as
was
well?
only like maybe 30 samples, but they were kind of a global.
Oh, they
global
were,
sampling.
okay.
And it was pretty immediate right at that point that, wow, this epidemic, this global epidemic is caused by a very small genotypic diversity. And And so there's an unusual genotype that's associated with most of these amphibian declines.
Oh, so it's rather specific then. And do you find Batrachochytrium that is different out in the wild?
Right. There, so for a while, maybe until about 2010, there was nothing known except this one genotype and all the sort of recombination variants it produces. But then people started discovering different genotypes, and one of them was a genotype that we discovered in Michigan. And this was a genotype that was on a bullfrog that we bought at a market.
In Michigan?
In Michigan.
I
And it
didn't
was
know they sold them in the Michigan markets.
They sell them everywhere, but not that many of them. It's kind of not a very thriving market.
Okay.
But you can
Boutique
find them.
market.
Yeah. People like their live fish and their live frogs. And so we isolated this strain. It was really unusual. So knew that, okay, there's a lot more diversity out there than we realized. And then we wanted to, we were working with Joyce and Lisa Schlegel, who was a PhD student with Eco Health Alliance or something like this.
Okay.
And her project was to understand the role of bullfrogs in spreading the disease globally. And so we understood that there are, if you buy a frog that's alive in a bullfrog for consumption that's alive in the U.S., it came from another country.
Oh.
Because there's no market, it's not lucrative enough.
Unless you live in Rain, Louisiana, the bullfrog capital of the U.S.
Oh, really?
Did
Okay.
you know about that?
No, I did not.
Okay, it's an incidental thing.
Yeah, so maybe there they'll sell them locally.
I think they do.
Yeah. They're not that bad. They're not that bad on the plate.
So it's easy then to buy live frogs. Do they ship them live or
They
frozen?
do,
Okay.
and that's the problem, is that because they're alive, they can have disease.
Mm-hmm.
And so we genotyped the frogs, and we more or less figured out with some certainty that the frog came from Brazil.
Mm-hmm.
And so we then thought, okay, there is this novel diversity in Brazil, which kind of makes sense. Brazil is it is like the most diverse country for amphibians. And so we started researching down in Brazil.
So is this the same species as we have here, Catesbiana? Is it right?
Yes, because they basically take the North American species, which is an aggressive invasive, and they farm that.
So it's been spread around then?
It has been spread around, but it's been spread around because it grows so well for the farming, and also when it gets into the wild, it's a big problem.
A couple years ago, before the last mycology class I taught probably, I phoned some dealers in Florida and asked them if they had any African frogs, and they said, oh yeah, we get a shipment, once a month. But if you want the ones that are raised here, you can have that too.
Right.
But there are apparently no rules at all, the guy said, for bringing in these, and I don't know if it's still that way or not. But I don't know who they were buying for. It may have been like a pet trade or something. Yeah.
Yeah, that should stop, really.
Yeah,
because
I just couldn't believe it was still going on. Now, recently there has been one, well, they've been finding it in Asia a lot, where it had hardly been collected in Asia before, but now it's thought to be the origin.
The center of origin,
Yeah.
yes. It's hard to collect there because it's the animals that have it are not heavily infected. There's no declines in die-offs because of the disease. So there were some isolates that were hard fought, and those were genotypically very distinct. They have high diversity, sexually recombining, and so Matt Fisher's lab spent a lot of effort putting together this huge team, and it's a strong hypothesis that it's an Asian origin of this global genotype, this genotype that's actually really related to the declines.
Yeah,
of
the one that
amphibians.
you find all over.
Yeah.
And so, is it hard to get permits to do this work?
Well, the animal work is harder.
Yeah.
So you need permits,
And they're
especially
vertebrates.
they're vertebrates. We're doing some stuff in the lab in Michigan where you need lots of permits or you know, whatever you have your protocols in place to deal with the animals. But working in Brazil, it's impossible without good collaborators. So the collaborators line up all the permits.
Yeah. So did you do a postdoc?
I did. So I was at Duke University for 9 years. I loved it so much in Rita's lab that I stayed for 3 more. And then during that time, I was on the Assembling the Fungal Tree of Life team.
Right, I kind of knew that.
Yeah, okay. That was my first postdoc. That was fun. I mean, Rita's lab was just a blast. There were so many mycologists that came through, both as students, visitors, etc. It was just a great environment.
Yeah. And then, from Meredith, you went to Michigan.
No, from there, actually, I went to Uppsala.
Oh, yeah.
And I spent a little over a year working with Hanna Johannesson and Jan Stenlund. And it was a great environment, and just had a lot of fun working with Hanna on Heterobasidion. And we were looking at how in a mycelium, which doesn't have sort of a regular nuclear control over the dikaryotic state, it's not dikaryotic, it's really heterokaryotic. So you have dozens of nuclei of one type and dozens of another type in the same mycelium, but they could have very different nuclear ratios, and you can actually modify those ratios by changing the medium, for example.
It's very simple. Okay, so then from Uppsala.
There's one
there's
more.
Another place.
Well,
Okay.
but I spent 1 year working with Jianping Xu in Master University, and he's a student of Jim Anderson, but also, so he's into mushrooms and medical mycology. I worked on Cryptococcus a little bit there. It was a very brief time. And then I got hired in my position at Michigan when I started in 2009.
And is that pretty much a dream job?
Yes, in a lot of ways. In a lot of ways, you know,
Yeah.
I couldn't ask for more in terms of the history, the support. It's fun. I'm asked to do what I always want to do, more or less.
You have pretty much freedom then,
Yeah, pretty much freedom.
as long as you get grant funding and students. Well, I visited there because I was at Hope College in Western Michigan for the first 6 years of my career. And I used to go over to use the library.
Who was there when you were going over there?
Frederick Sparrow.
Okay, wow.
He was the mycologist there. And he, I didn't dare to go in and introduce myself and talk to him when I was using the library, but he actually saw me and recognized me from meetings, and was just wonderful because he told me, you don't have to drive over here. I will send you anything you need. He was going to photocopy it and send it. And I've always wondered, he took me to his office, and he had a letter that had been addressed to Millardet, who did the Bordeaux mixture. And he had bought it somewhere, or someone had given it to him as a souvenir of that time. And I always wondered after he died what happened to that letter. It was the envelope, maybe only the envelope.
Okay.
But it was sort of really nice to see this and have it connected with fungi, and Bordeaux mixture was used out in the grape fields first. But and I guess Millardet was a Bordeaux professor who started using it, developed the Bordeaux mixture. But anyway, he was just a lovely person. And the mycologist after him was Vogel.
There was Vogel and there's Bob Shafer.
But Shafer was there too, I think.
Shafer was there when you were there.
Yeah, and he's a really nice person too. Several people have been influenced by him, if not actually taught by him.
And he still tries to have his mushroom hunting club that goes out on the weekends in the fall and gets together, collects mushrooms, and eats together.
Linda Cohn and Jim Anderson were talking about him, and then also at this meeting, the Mendocino foray, Tom Bruns talked about his labels that he had.
Right.
Yeah.
He
Yep.
still has them. He
So
still
it's
uses them.
still going on.
Yep.
Good to know.
Yeah.
So at Michigan, you've worked in several really nice areas, your research areas, and your website reflects all this. It's a nice website.
Oh, thanks.
And so how many postdocs, do you have postdocs now? And
Yeah, I have 2 postdocs. One of them is Rayburn Simmons, who was
yes.
Joyce Longcore's PhD student, and I also have in there Kensuke Sato. He was a student of Yusuke Degawa, and both of these guys are Kittredge guys, and they're amazing at what they do in terms of natural history and systematics of Kittredge.
do you do any microscopy, electron microscopy with the Kittredge, or are they characterized already, the ones you're looking at?
We have done some electron microscopy there in Michigan, not a whole lot. The field is not moving away from that, but it is very laborious, as you know.
It's very laborious. Yeah, I guess you have to be able to cut serial sections.
Right. Yeah.
Someone I know has been trying to find someone to cut serial sections, and the places where she's inquired if they could do it, they don't really know what serial sections
really?
are. I was surprised, but yeah, microscopy I think is still important. And so what are your students doing?
Whatever they want to do,
Okay.
honestly.
That's good.
We have a good program there that really sort of allows the students to do anything in mycology that's evolution ecology related. So I encourage them to come up with their own projects.
That's good.
And that's why you have, a student working on mushroom methylation during development, a student working on mycoviruses of basal fungal lineages, a student working on soil ecology and wood decay ecology, and there are others. So
It's pretty exciting.
it's fun to have all these projects going on. Sometimes it's a little bit hard to pull things forward, and there's less synergism sometimes than I would like to have, but it's also exciting to see students, develop their own ideas and bring them to fruition.
Yes. So is there anything else that we haven't talked about? That you
Well, I think maybe the last thing I could talk about was that, I've had these great collaborations with so many people, but I'll just mention that I've mentioned Joyce multiple times. But also, we've got this new culture collection. I don't know if you saw the poster on it,
No.
but it's called the Cesium Collection, and it stands for Collection of Zoosporic Eufungi at the University of Michigan. And so we have over 1,000 isolates of these basically Kittredge and blastoclads, and we've taken Joyce Longcore's collection, this thing that she built over 30 years, and also we now have Martha Powell and Pete Letcher's collections, and we're basically safeguarding those for the future and putting them out there as a resource for the community that's interested in studying Kittredge.
Yeah.
The work that they put into generating these could never be replicated, so our effort will be in maintaining that record.
Yeah, and it's also very, very expensive, especially if you go far afield to collect these things, even if they're not zoosporic things, because those probably require even more work. But just trying to collect yeast worldwide is a difficult problem. Yeah, so that's great you're doing that. I had no idea. We were talking about yesterday, someone I know who's retiring didn't know what to do with his collection.
What kind of fungi were they?
They were Aspergillus penicillin. You want to take those on too?
We'd have to have a different
But
different collection.
I'm sure there are places that would do it for those. Yeah, yeah. Well, Tim, thanks. It's been great to talk
Yeah,
to you.
it's nice to talk to you too, and this is a wonderful project. So
Well, I
I'm looking
hope
forward to
it works. And so we've got about 33 people that we interviewed, in some of them multiple people in an interview, but
Right.
not too many. And so I think we've had maybe up to 12 or so this time. And
Great.
it's a shorter meeting than Puerto Rico was,
Mhm.
and so we may not get so many, but it's really good. We're getting different people who didn't go to Puerto Rico, like you, so that's
Right.
great.
Yeah.
Okay, well,
Yeah. Thank you, Meredith.
thank you, Tim.